Good morning, everyone. Today we will talk about the study of media, the media which is used in the fermentation process. In fermentation process, the choice of the most optimum microorganism and fermentation media is very important for high yield of product. The quality of fermentation media is important for microorganism. The media provides substrate for product synthesis in a fermenter. So basically, fermentation media is of two types, major and minor. In major components, it contains carbon nitrogen source, and in case of minor component, it contains inorganic salt, vitamins, growth factors, antifoaming agent, buffer, dissolved oxygen, and other dissolved gases, growth inhibitors, and enzymes. Nutrients required for fermentation media also depend on the type of fermentation organism as well as on the type of fermentation used. The ideal characteristics of fermentation media should meet the following criteria. Now, all these ideal characteristics should be contained in a media for the optimum fermentation process. Number one, production of the maximum yield of product or biomass in short duration, a minimum yield of undersized other products, free from toxic effects on culture or product. It should be of low cost, low quality, and easily available. Production medium should be suitable for adjustment of pH, consistency, and composition. Foaming must be minimum. Media components must not interfere with the extraction and purification of the desired product. More suitable for production process such as agitation, extraction, purification, etc. Basically, two types of media, two types of fermentation media are uses, growth media and fermentation media. Growth media, it contains low amount of nutrients. It is useful in creating raw material for further fermentation process. Fermentation media contains high amount of nutrients. It is used in creating final products using fermentation. For example, growth of yeast requires 1% carbon, but during fermentation of alcohol, yeast require 12 to 13% of carbon in the medium. So there are two types of fermentation media which is used in the industry. First is synthetic media and another one is crude media. In synthetic media is used in the field of research as each and every component is chemically known and exact composition of nutrients is predetermined. So, in case of synthetic media, variation in levels and concentration of nutrients can be controlled. So, synthetic media is having advantages as well as some disadvantages also. Talking about its advantages, it lacks source of protein and peptides, hence there is no formation of any foam. Another advantage is it is useful in controlling the growth and yield of products in lab. Talking talking about the disadvantages of synthetic media, the main thing is the cost of the media. It is more expensive. The most important aspect of fermentation is that it should be economical and profitable. That's why synthetic media is not used in industrial scale. Another one is crude media. Crude media is generally used on an industrial scale for fermentation process as it is low cost media. Crude media contains a rough composition of media required for fermentation. It gives high yield of product and contains undefined source of ingredients. Crude media contains high level of nutrients, vitamins, proteins, growth factors, antifoaming agents and precursors. Discussing about the ingredients of crude media, it contains inorganic nutrients. Crude media contains inorganic salts containing cations and anions along with a carbon source. Sometimes fermentation microorganisms have a specific requirement of ions like magnesium ion, phosphate or sulfates. These requirements are fulfilled by the addition of these ions to balance the crude media. Another is carbon source. The common carbon source used in pharmaceutical fermentation process are glucose, fructose, sucrose, molasses, hydrolyzed starch, organic acid, hydrocarbons, plant oils, etc. Blackstrap molasses is the cheapest and most useful sugar source for industrial fermentation. The another source is nitrogen source. Salts of urea, ammonia, and nitrate can be used as nitrogen source when fermentation organisms are non-proteolytic in nature. Pure from urea, ammonia, and nitrates are used as a source of nitrogen. Another one is growth factors. Crude media constituents provide enough amount of growth factor, so extra addition of growth factor is not required. Another one is buffers. Buffers are used to control the pH changes in order to maintain the pH throughout the fermentation process. Many buffers have been used in the process of fermentation. For example, proteins, peptides, amino acid act as a good buffer at neutral pH. There are other inorganic buffers also like calcium carbonate, potassium hydroxide, which are easily available buffers. The another part is precursors. Precursors are generally present in the media as crude constituents. Precursors are added in the fermentation media at the time of fermentation as it get incorporated in the molecules of product without bringing any change to the final product. So basically, what is the use of precursors? Precursors helps in improving yield and quality of the products. So basically, precursors are added in the pure form depending upon the need of the product. It depends upon the type of our process, on the type of our product. For example, chloride is added less than 10 ppm in fermentation of vitamin B112. As in my previous videos also, I have explained the fermentation process, the production of vitamin B112, and there in the process of vitamin B12, cobalt chloride is used as a precursor to enhance or to improve the yield. Now the another one is antifoaming agent. Fermentation media containing proteins or peptides. It can produce foam in the process of agitation and aeration. Proteolytic bacteria mainly produce high level of foam. If this is not controlled, it may give rise to head space of the tank. This condition is mainly responsible for causing contamination of the fermenter. So because of this contamination, we need antifoaming agents. Antifoaming agents can lower the surface tension and decrease the stability of foam bubbles. Commonly used antifoaming agents are corn oil, soybean oil.